Hi everybody, I'm Dr. Biomed. Welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified, and summarized form. This video is about the structure of lipids in the cell membrane. Please subscribe to this channel, and if you like the contents of this video, please like it. First of all, I recommend watching my previous video. Its name is the classification of cell membrane components, and you can find its address in the description. After watching that video, you will know the accurate classification of cell membrane components. Okay, now I want to speak about phospholipids group. Lipids in the cell membrane are in three groups. The first group is phospholipids. The first subgroup of phospholipids is phosphoglyceride, which are glycerol-based phospholipids. Phosphatidyls are a subgroup of phosphoglyceride. They have two parts in their structure: hydrophilic head or polar head group and hydrophobic head or nonpolar head group. The polar head has three components: glycerol as a backbone, phosphate, and head group. And nonpolar head group has two fatty acid tails. Fatty acid linked to glycerol by ester linkage and phosphate linked to glycerol by phosphodiester linkage. Fatty acids are different from each other. These differences are the length, that can be between 14 and 24 carbons, and saturation. Fatty acids can be saturated or unsaturated. Saturated means there are no cis double bonds, and unsaturated means there are one or more cis double bonds. The effect of differences in fatty acid tails is the fluidity of the membrane. Okay, phosphatidyls have many types. Their structure are different from each other in the head group. One of them is phosphatidyl ethanolamine. It has two parts: hydrophilic head or polar head group and hydrophobic tails or nonpolar head group. The polar head contains glycerol as a backbone, phosphate, and ethanolamine. Nonpolar head group contains two fatty acids tail. Fatty acids linked to the glycerol by ester linkage, and phosphate linked to glycerol by phosphodiester linkage. The, char- the charge of phosphatidyl ethanolamine is neutral at physiological pH, carrying one positive and one negative charge. It's located in mammalian cell membranes, particularly in nerve cells and in bacteria. It plays a role in membrane fusion, cell division, and precursor and substrate in biological pathway. Okay, phosphatidyl serine. Phosphatidylserine has the same structure except for the head group. It has hydrophilic head or polar head group and hydrophobic tails or nonpolar head group. Hydrophilic head contains glycerol as a backbone, phosphate, and serine. And nonpolar head group contains two fatty acid tail. Fatty acid tail linked to glycerol by ester linkage and phosphate linked to glycerol by phosphodiester linkage. Phosphatidylserine carries a net negative charge. It's located in mammalian cell membranes and it plays a role in cell cycle signaling, specifically in relation to apoptosis. Okay, phosphatidylcholine. Like other phosphatidylcholine has two parts. The only difference is the head group, which is choline here. Ester linkage and phosphodiester linkage is the same. It's electrically neutral at physiological pH, carrying one positive and one negative charge. It's located in mammalian cell membrane and it plays a role in membrane-mediated cell signaling and PCTP or phosphatidylcholine transfer protein activation of other enzymes. PCTP transfers phosphatidylcholine molecule between membranes in vitro. Phosphatidylinositol. Phosphatidylinositol has the same structure as other phosphatidyls, except for inositol. It carries 4 negative charge, is located in mammalian cell membrane, and it plays roles in cell signaling and lipid signaling. And phosphatidylglycerol. Phosphatidylglycerol has the same structure except the head group that here is glycerol and the same linkage. It carries a net negative charge located in mammalian cell membrane and plays a role in activation lipid-gated ion channels. Okay, another subgroup of phosphoglycerides is cardiolipins or the phosphatidyls glycerol. In their structure, they have two parts too. Hydrophilic head or polar head group and hydrophobic tails or nonpolar head group. Polar head group contains glycerol as a backbone, 2 phosphates, and 2 glycerol. And nonpolar head group contains 4 fatty acid tails. Linkage is the same. They carry 2 negative charge. Their location is in the membranes of most bacteria and the inner mitochondrial membrane. It plays a role in bacterial and mitochondrial energy metabolism. Another important class of phospholipids is the sphingolipids or ceramide, which are built from sphingosine rather than glycerol. They have 2 parts in their structure: hydrophilic head or polar head group and hydrophobic tails or nonpolar head group. Hydrophilic head contains phosphate and head group. And nonpolar head group contains a sphingosine as a backbone and one fatty acid tail. Fatty acid tail linked to a sphingosine by amide linkage, and phosphate linked to a sphingosine by phosphodiester linkage. They have a role in signal transduction and cell recognition. Sphingomyelin is the most common sphingolipid. A fatty acid tail is attached to the sphingosine with an amide linkage and phosphocholine with a phosphodiester linkage. It is located in animal nerve cells and it plays a role in signaling pathway and cell apoptosis. Another subgroup of sphingolipids is sphingosine. Sphingosine has two parts in its structure too: polar head group and non-polar head group. Polar head group contains two OH groups, and non-polar head groups contains a sphingosine as a backbone and one fatty acid tail that it's unsaturated. Fatty acid tail linked to a sphingosine by amide linkage. It's located in animal cells and it plays a role in signaling pathway, apoptosis, cell cycle arrest, and the precursor of more complex sphingolipids. The second group of lipids in the cell membrane is sterols. Animals' plasma membranes contain a specifically large amount of cholesterol. Cholesterol structure. It has hydrophilic head or polar head group for fused ring structure and hydrophobic tails or nonpolar head group. Polar head group contains hydroxyl group in the A ring for fusing structure A, B, C, and D, and nonpolar head group contains one fatty acid tails. It's located in a plasma membrane of animals. It plays a role in maintaining membrane fluidity by preventing fatty acids chain from coming together. Precursor of testosterone, estradiol, and vitamin D, and thicken the cell membrane. The cholesterol molecule orient themselves in the bilayer with their hydroxy group close to the polar head groups of adjacent phospholipid molecule. Plant plasma membrane contain large amounts of ergosterol. Its structure has three parts too: hydrophilic head, ring structure, and hydrophobic tail. Hydrophilic head has hydroxyl group. Ring structures have A, B, C, and D fused ring, and nonpolar head group has one fatty acid tail. Location of ergosterol is in a plasma membrane of yeast, fungi, and protozoa, and in human nutritional compounds. Role of ergosterol is precursor of vitamin D. Ergosterol has two additional double bonds in comparison to cholesterol. One double bond in a ring and another double bond in fatty acid chain. The third group of lipids in cell membrane is glycolipids. Sugar-containing lipid molecules are called glycolipids. They resemble sphingolipids, but instead of a phosphate link head group, they have sugars attached. Okay, glycolipids have two subgroups. One of them is glycosphingolipids. Hydrophilic head of them has carbohydrates, and hydrophobic tail has a sphingolipid as a backbone and one fatty acid tail. Carbohydrate linked to sphingolipid by glycosidic linkage, and fatty acid tail linked to sphingolipid by amide linkage. They are located in animal cell membrane, and they play a role in cell adhesion, oncogenesis, and cell-to-cell interaction. In order not to get confused, I recommend you watch my previous video about the classification of cell membrane components. Okay, glycosphingolipids are divided into two subtypes. Cerebrolipids or cerebrosides is one of them. Same as the rest, they have two parts in their structure. Hydrophilic head has monosaccharide, glucose or galactose, and hydrophobic tail has sphingolipid as a backbone and one fatty acid tail. Monosaccharide and fatty acid tail link to sphingolipid by glycosidic linkage and amide linkage. It's located in animal muscle and nerve cell membrane, and it plays a role in cellular recognition and cell-to-cell communication. Glucocerebroside or glucosylceramide, which is one of the cerebroside subgroup, has glucose as a polar head group and a sphingolipid as a backbone and one fatty acid tail as a non-polar head group. It's located in brain and in peripheral nerve system, and it plays a role in cellular recognition and cell-to-cell communication. Okay, galactocerebroside, the second subgroup of cerebrosides, has galactose in the polar head group and a sphingolipid and one fatty acid tail in non-polar head group. It's located in neural cells, and it plays a role in cellular recognition and cell-to-cell communication. The second subtype of glycosphingolipids is ganglioside. They are complex and contain oligosaccharide with one or more sialic acid. And a sphingolipid and one fatty acid tail as non-polar head group. is located in ganglion cells and in CNC. It plays a role in cellular recognition and cell-to-cell communication. The second subgroup of glycolipids is galactolipids. Unlike glycophospholipids, galactolipids have glycerol as a backbone. Glycerol and galactose make hydrophilic head, and two fatty acid tails make nonpolar head group. Galactose linked to glycerol by glycosidic linkage, and fatty acid tails linked to glycerol by ester linkage. Their location is in plant cell membrane, and their role is in photosynthesis. Galactolipids are divided into two categories. One category is monogalactosyl deacylglycerol or MGDG, which has one galactose in the polar head. It's located in chloroplast membranes and thylakoid, and it plays a role in photosynthesis. Another category of galactolipids is D-galactosyl deacylglycerol or DGDG. As you see, it has two galactose in polar head group. Its location and role are the same to MGDG. Thank you for watching. Please give me a comment and tell me what topics you would like to be presented in this cafe. In the next video, I will talk about the structure of proteins in a cell membrane in a simple, classified, and summarized form. Until the next video, bye!